afternoon, ladies and gentlemen. I also from the Jiangnan University. Our university is in Wuxi, China. It's very close to Shanghai, just 120 kilometers. And I will report 4 parts about our research. The first part is the application of the α-keto acid. The type and the structure of α-keto acid. Some typical α-keto acid are some, for example, the α-keto glutaric acid and α-keto isocapric acid and similar. And the application of α-keto acid. Α-keto acid find a wide application in food, medicine, health, and chemical synthesis. For example, in food industry. Can be used substitution for amino acid as food, and also can improve the flavor and the taste, and also can be used in the medicine and beauty and health, also chemical synthesis. And the comparison of different production methods. For the production method for α-keto acid, there are usually 3 methods. One is chemical synthesis. Chemical synthesis process is the main method to produce keto acid till now. The advantage is the short reaction path and the high conversion rate, but there are also some disadvantages for this process. For example, the toxic catalyst, high temperature and pressure, and also energy consumption. And the second method is a microbial fermentation process. And some keto acid can be produced by this method. The advantage is the recycle of the raw material and the low pollution. But there are also some disadvantages. For example, the complicated metabolic pathway and the low conversion rate with this method. And the third one is by transformation process to produce the alpha-keto acid from amino acid by using L-amino acid deaminase, abbreviated as LAAD. And this one can be, this enzyme can be used to produce several alpha-keto acid. The advantage is a high conversion rate and simple reaction system, and also low purification cost. But there are also some disadvantages with this method, including the very low, or not very high production because of the low substrate tolerance. The third part is by transformation production of α-keto acid. I will give 3 examples to introduce our research work. The first one is the biotransformation of glutamate to α-KG. α-KG is a very important alpha-keto acid, and also there are some functions in body reinforcing, can be used as a body reinforcing agent and additives of drinks, and also some other applications. For the synthesis of for the synthesis of alpha-keto acid, there are several ways. One is the chemical synthesis, but they are including several parts. But for biotransformation, there are only one steps. But currently, alpha-KG was mainly produced by chemical synthesis. For biotransformation process, there are some advantages. For example, one step and high efficiency, no toxic catalyst, and less pollution. And in our research, we express the gene of LLAD from this Proteus myelobialysis from this gene, and the gene was expressed and the protein was purified, and but it's expressed in the E. coli as an inclusion body, so we did the refolding of the encroaching body, and the active protein was obtained. And this is the optimization of the biotransformation process with this enzyme. And we optimize transformation condition. And the conversion rate is not very high. And 1.5 gram per liter of α-keto can be produced. Compared with free enzyme system, the wholesale biocatalyst have some advantages. So we expressed this enzyme in E. coli and bacillus, and did the biotransformation with the wholesale system. And by optimize the biotransformation condition, the α-keto, α-kg titer could be improved to 4.6 gram per liter. And we also did some directed evolution and side-directed mutagenesis to this enzyme. So through the 3 rounds of allo-prone PCR and also the side-directed mutation, one of the optimal mutant was obtained. And with this mutant, the conversion rate and α-kg titer could be improved to 8.6 gram per liter. And also, by the deletion of the α-kg dehydrogenase to reduce its degradation, the α-kg titer could be further improved to 12.2 gram per liter. And through the gene shuffling and allo-prone PCR of this enzyme, the biotransformation rate was further increased. This is the strategy for the evolution of the L-amino acid deaminase. And in Flask, by optimize biotransformation condition, the α-kg titer could be improved to 52.6 gram per liter. And the biotransformation also was conducted in 3-liter fermenter, and the conversion rate and also the α-kg titer was improved by using the feedback culture system. Second example is the biotransformation of L-phenylaniline to PAA. PAA is widely used in pharmaceutical, food, and chemical industry. And the gene also from the Proteus mylobalis was expressed and induction condition was and the purification step was optimized. These are some detailed condition, and the enzyme were purified, and the overall yield is 13%. And with this enzyme, we did the enzymatic biotransformation from the phenylaniline to PPA, and the maximum conversion rate and PPA titer reached 86.7% and 2.6 gram per liter by the reaction of 2.5 hours. And by using the wholesale transformation system, the maximum conversion rate and PPA titer was further improved. But the reaction time was elonged, so it's needed longer reaction time with the wholesale system. Through the metabolic engineering to delete the PPA degradation pathway in E. coli, we did the single, double, and trip deletion, and several mutant was obtained, and the final result with for the trip deletion mutant, the PPA titer reached 3.9 gram per liter, and also directed evolution and side-directed mutation of AIDD was did through the 2 rounds of allo-prone PCR, and also the side-directed mutation. We get the optimal mutant, and with this mutant, the PPA titer reached 10 gram per liter, with a conversion ratio, with a very high conversion ratio, almost 100%. And also the kinetic analysis showed that the triple mutant had a higher substrate binding affinity and catalytic efficiency than the wild type. And the feedback by transformation in Flask was did by feeding the substrate every hour. The maximum PPA production reached 21 gram per liter. And also the effect of the PPA addition on initial rate was conducted to determine the effect of PPA addition on the initial rate, also to determine the product inhibition constant during the biotransformation process. So it is determined by kinetic analysis showed that it is a kind of competitive inhibition biotransformation process. This is the model. And the general rate reaction for PPA production model was obtained, and also the consumption of the substrate model and consumption of the Biocatalyst model was obtained. And through the experimental determination of the rate constant, some constant in the model was obtained. The substrate and the production inhibition of the engineered E. coli was 65.8% and 68.1% of this the white type strain. The biotransformation in 3-liter fermenter was did by the wholesale biotransformation in 3-liter fermenter with different concentration of the substrate. The conversion rate reached 100% within 4 hours, and no substrate inhibition was observed with 35 gram per liter of the substrate, and also the improved dissolved oxygen can speed up the biotransformation process and increase the productivity. The third example is the biotransformation of leucine to α-keto isocapolate. This α-keto acid is also widely used in pharmaceutical, health product, and feed. This α-keto acid can be produced by the transformation of leucine with also the L-amino acid deaminase by expressing the gene from this one in E. coli. And we also optimize the production by the wholesale system, and by optimize the condition, the conversion rate can reach the 97.8% with the substrate of 13.1 gram per liter of the substrate. And also, we also did determine the effect of the air-reducing transporter on the wholesale biocatalyst activity by addition of the CCP. CCP is the chemicals. When this one was used to uncouple the agent that catalyze the electrogenic proton movement to reduce the ATP generation and inhibit the transporter efficiency. CCCP also can improve the biocatalyst activity at the some concentration. When the substrate concentration below 20 millimole and the highest KIC production reached 47.1 gram per liter. And also effected of the different substrate supply strategy was used to improve the KIC production. Batch and interval using feeding on the KIC production was studied in Flask. By comparison, batch and feeding of the substrate, in feeding process, the very high KIC titer could be reached. And also, we also did some research to improve the LDD activity with a different plasmid copper number. This is the table listed the different plasmid copper number, 10, 5, or the highest one is 100. And the results showed that the cell growth rate increased with decreased plasmid copper number. But with 10 copper number, the KIC can reach the very highest value, 76.5 gram per liter. And we also improved the expressing of the enzyme by different, this also compared the different plasmid of the iron level, cell growth, and KIC production. The results show the higher plasmid copper number do not result in the higher iron level, which means the transportation of the enzyme is limited at the higher plasmid copper number. And the expression of the AIDD was improved by comparing the different N-terminal condom base and RNA structure. The results showed that some mutant can get, with some mutant, the higher relative activity could be reached, and the highest KIC production titer could reach the 79.7 gram per liter, and also by changing the synthetic ribosome binding site, the protein to control the protein expression, and the highest KIC production can reach the more than 80 gram per liter. This is summary of our research, and we express the gene from the different strain, and also by increase the catalytic activity by using different strategy, and also optimize by transformation condition the alpha-keto acid, different alpha-keto acid production could be improved by the different strategies. So this is my presentation. Thank you. Thank you very much. Oh, okay. Yeah.
What are the next steps? Are you going to develop new processes for all the alpha-keto acids?
Yeah. We developed more than 3 process, but some production title also still need to be improved before using in the industry. Yes.